Hello, Professor. This is Emiliana, and for my panel video presentation, I will be doing the Angelman syndrome. So, what is the Angelman syndrome? It is a genetic disorder that primarily affects the nervous system and is considered a rare neurogenetic disorder. It is estimated to affect a 1 in 2,000 to 20,000 people. Characteristics are a delayed development, intellectual disability, a severe speech impairment, problems with balance, a small head size, difficulty sleeping, and some additional facts are that the syndrome is mainly developed at the beginning of the ages of 2 to 3 years old, stiff of jerky movements when walking. There is a small size of the head with a flatness in the back of the head than the tongue thrusting, and normally the hair, skin, and eye colors are light color. Um, and there's an unusual behavior when walking. You can see in picture above, picture D. There's a flapping and hands flapping and arms up lifted when walking. Okay, what causes the Angelman syndrome? There's different ways of causing AS. So, normally it is the loss of function of the gene UBE3. So, people normally inherited one copy of the UBE3A gene from each parent. Both the copies of these genes are turned inactive in many of the body's tissue. In certain areas of the brain, however, only the copy inherited from a person's mother is activated. This parent-specific gene action is caused by a phenomenon called genomic imprinting, and if the maternal copy of the gene is lost because of a chromosomal change or a gene mutation, then there's no action copies of the gene in some part of the brain, so causing the syndrome. There's also several different genetic mechanisms can inactivate or delete the maternal copy due to occur when a segmentation of maternal chromosome 15 can be deleted. So that's roughly 70% of the chances, and 11% it is caused by a mutation in the maternal copy of the UBE3A, causing the Angelman disorder. In addition, there's a small percentage that is caused by uniparental disomy. This is when the father or when a person inherits both chromosome 15 from one parent, when in actually they supposed to have each chromosome from both parents. This caused a this could cause the Angelman syndrome, and it is caused by a chromosomal arrangement called a translocation or by a mutation or other defect in the region of the DNA that controls the actions of the UBE3A gene. So these genetic changes can abnormally turn off or inactivate the UBE3A or these genes on the maternal copy of the chromosome 15. So for the or other causes are the loss of the gene OCA2, which causes light color hair, what I mentioned before. So a it's normally a characteristic of the syndrome, and this gene is located in chromosome 15, which sometimes, as I mentioned before, in the other causes, it can be or deleted or create a mutation, and this is why the Angelman disorder is created. And roughly about 10 to 15% of the cases are still unknown why there is that there's not been determined any cause, and research is still being done. A little bit about history of this syndrome. Okay, so starting 1965, they discovered AS, which was Dr. Henry Angelman, which is an English philosopher, published a research paper about AS and some characteristics of children with this syndrome. The diagnosis of AS in 1928 with Dr. Williams and Dr. Frias were the first patients that could diagnose this syndrome, and by 1987, genetic marker was identified. So Ellen McGuinness, physician at the Oregon Health Science Center, discovered the genetic markers for AS, a missing genetic code as a tiny portion of the chromosome 15. For the treatment, there's usually no cure for the Angelman syndrome, but it's still being researched. So some treatments are anti-seizure medication to control seizures, physical therapy to help with walking and movement problems, communication therapy including sign language and picture communication, behavior therapy to help overcome hyperactivity and short attention span and aid development. And how to determine if the person has a Angelman disorder. So there's a testing process. So if by a DNA by the obtained DNA from a blood test, there's the methylation test results, which can be normal or abnormal. If it's normal, then the UBE3A gene is tested, and if it's normal, A is unlikely to still be possible. There's a 10%. But if it has a mutation in the UBE3A, then it is more likely to have a Angelman disorder. But if the methylation test result is abnormal, then there are three mechanisms are possible for chromosome deletion. So one is or a deletion is by a UDP or imprinting defects. So fishing or CGH study, then there's chromosomal deletion. There's 70% of all with a yes. There's normal DNA marker, paternal uniparental disomy, and imprinting defects is presumed since a chromosome deletion and UPD has been excluded. Molecular studies of imprinting sensors can be determined if it has a deletion in it. So the chromosome deletion and the paternal uniparental disomy and the imprinting defects are also three other ways to understand the chromosome deletion or the imprinting defect. Here is an additional video that I added to the presentation. It's showing it's showing a little bit of how does Angelman look like and how people live with this syndrome day to day.
Just as every human being is unique, so too is every person born with Angelman syndrome. It was once believed that to have Angelman syndrome, you had to have blue eyes and blonde hair. But you might find that some also have dark hair, brown eyes, and the skin can come in olive. Jada is an Aquarius and curious. Billy was born in the year of the rooster and has a keen sense of pride. Mendy is Jewish and Tara is English. Charlotte has strabismus. Lucy has wider spacing in her teeth, but Hannah doesn't have either of these.
The video goes on, but this is a little demonstration on how Angelman syndrome is normal and you can live with it even though there's no cure. So this is the end. Professor, thank you for everything and have a nice summer.